At hLab, we predominantly focus on infectious diseases, and we are specialized in providing support and expertise in the field of virology, microbiology, and immunology, from a preclinical level up to the clinical trial levels.
Traditional system takes about 3 hours. The new generation of robotic system for extraction requires simple like half an hour. So that brings the possibility for people to expand the throughput of the system. We can manage more samples in less time.
Because the sensitivity, the scalability, the reproducibility within the system, within the QX700, it helps us accelerating research, and it helps us cutting the turnaround time of discovering a new target. It will allow us to move very fast at pace with confidence throughout the early stages of the drug discovery. And then when you are at the level of a clinical trial, you can have a trusted data output quite fast in a very accurate manner. So the NGS offers a landscape. The digital PCR verifies and quantifies the findings in the NGS.
The addition of the QX700 to hVIVO is an important addition. We absolutely want to be a differentiator and be seen to be bringing new technology and new applications forward within the vaccine and ultimately the drug discovery space. Infectious disease is only going to be an increasing burden on health systems, an increasing health issue for the human population. So having a proactive approach is going to be really important. We have to understand that a lot of the work that we do involves and revolves around clinical samples which have been generated from heavily invested and expensive clinical studies. Those samples are a defined kind of volume and obviously very valuable in terms of what they potentially can show. So when it comes to using the platform and the instrumentation, we are looking for accuracy, robustness, relatively low sample volumes, but maximising the information that we can get from those samples. And I think the sensitivity, the quantification aspects, but also the multiplexing aspects of this particular platform is really advantageous for us.
When you run a clinical trial, it's always like patient safety in mind. So you really need to work with one small volume, one small sample, one small clinical testing that will allow you to get the most out of it.
Using volumes like 5 microliters, a drop of liquid targeting 21 up to 28 different targets brings a lot of possibilities to interrogate problem in HR.